everyone, I am Dr. Manoj Kumar Sharma, Associate Professor, ITS College of Pharmacy, Muradnagar. This is the lecture number 24 and the topic is designing the methodology, and it is the first part of this topic, and this topic of the unit 3, and the course is Bachelor in Pharmacy, and the subject is Biostatistics and Research Methodology, and the code is BP 801T, and the university is Dr. APJ Abdul Kalam Technical University. The content we cover in this lecture: what is sample and what is the sample size and the power of a sample size. So, what is sample? Sample, it is the small representative part of the population. The sample is a small part of the population, but there is another word representative. That means, the sample should be representative of the population. The sample is a small part of the population and it contains all the characteristics of the population. Why do we use samples? We draw the sample or we use the sample, get information from large population with at a minimum cost and maximum speed, that means to complete our task in minimum time and to increase the accuracy. The main aim of use the sample is we want to reduce the cost of our research and increase the precision. These are the main 2 points to use the sample. To reduce the cost, to reduce the time to complete the study and increase the accuracy of the result. So, for these 3 objectives we use samples. And second point is why sample size calculation. So, if we have a large sample, it will require more time to cover study, and if it is a survey, if we have any survey study, then we need more person to cover the survey or more investigator to collect the information in the survey, and if we it will take more cost to complete the study or survey, as when we have more investigator, then we need more money, and if we want to cover more geographical area, then we also need more time, more person or investigator, and it will also increase the cost of our study. So, and as we know, the main aim in our to calculate the sample is we want to reduce the cost, we want to reduce the time to complete the study, and we want to increase the accuracy. So, when we have large sample, it will have more cost, more time and more investigator or more persons. And it may be unethical. So, and if we have small samples, then if we have small samples, then it may be some characteristics of the population may or be absent, and then the sample is not representative of the population, and also in small samples these are unable to detect clinically important result. So, there is a problem. If we have large sample, it require more cost, more time and more investigators or more persons, and if we take small samples, then it may be some characteristics are absent and the results are not good. So, and it is also unable to detect clinically important results. The sample should be not very small and not very large. I explained this in clinical significance in 1966. Colson et L had shown 25% reduction in mortality due to propanol among MI patient, but that was not significant. Significance, the changes are not real. When we say the result is not significant, then changes are not real, but they may be by chance at given level of probability. So, that was not significant. 66 cases and 64 controls were studied. So, is 25% mortality in reduction is important, not important. Why we say this? The worry is that the difference in mortality, that is 25%, could have occurred by chance alone. Okay? So, it is not significant, it is not real. So, if 25% is mortality clinically relevant, then why sample size calculation? Because the 25% difference is not significant as it occurred by chance. In order to provide the 25% reduction, mortality is statistically significant, they should have studied 200 patients in each arm, that is 200 control and 200 cases to get the result. So, this is the sample size for this study, and the sample size, when we calculate the sample size, it depends upon the types of medical research. As there are two types of medical research, first is estimation. In estimation, we find the prevalence of any disease, or we do descriptive study that, and in estimation study there are two conditions. First, either given proportion or prevalence, and second, we have mean and standard deviation. Another type of medical research is testing of hypothesis. In test of hypothesis, we have cohort or case control study, clinical trials, and in testing of hypothesis, we have two proportion or incidence rate of any disease, and also if we have two group and we want to compare them, then we have the two group means and standard deviation. So, there are two types of medical research, estimation and testing of hypothesis. In estimation, we find the prevalence of any disease, or we describe about the population characteristics, and to calculate the sample size in estimation, we have two things, either we have proportion or prevalence of any disease, and second we have mean and standard deviation of the data. And in testing of hypothesis, to calculate the sample size, we have two things, either we have two proportion or incidence rates, and second case we have two groups mean and standard deviation. Now, how we calculate the sample size? There are different formula. Now, first type of study is estimation. For estimation, the first condition, when proportion is the parameter of our study, then the sample size n equal to z square into p into q upon d square, where capital Z is the standardized normal deviate or z value, p is the proportion or prevalence of interest, q is the equal to 100 minus p, and d is clinical expected variation or precision of our study. For example, from a pilot study, it was reported that among headache patient 28% had vascular headaches. It was decided to have 95% confidence interval and 10% variability in the estimated 28%. So, how many patients are necessary to conduct the study? Here we have p, that is proportion is 28% and q equal to, as we know, q equal to p minus 100 minus p, and this is 100 minus 28%. So, it will equal to 72%. Z alpha for level of significance 5%, the value of Z is 1.96, this is the standard value from the Z distribution, and we remember if we have level of significance or alpha or first type of error is 0.5, the value z is 1.96, and in this the precision or clinical significance d equal to 10% of 28%. So, this is equal to 2.8. On putting these value, formula is n equal to z square p into q upon d square. When we put these values z equal to 1.96, p equal to 28, q equal to 72 and d is 2.8, we get the sample size is 987.8, and in round off we take 1000 sample. So, this is the sample size when the prevalence or proportion is interest, and it is the type of study, estimation study. Second case, when the mean is the parameter of our study, that we want to estimate the mean of any parameter, mean of any type of variable, or you can say when mean is the parameter of our study, we want to estimate the mean from of a population. For example, if we want to estimate the height, average height of a population, then the formula n equal to z square into s square upon d square, where capital Z is standardized normal deviate as in the previous formula, s is the sample standard deviation and d is the clinically expected variation or precision. For example, in a health survey of school children, it is found that the mean hemoglobin level of 55 boys is 10.2 per 100 ml with a standard deviation of 2.1, and consider the precision is 0.8, and what will be the sample size for this study. So, as in this example we have mean equal to 10.2, standard deviation is 2.1, and we know the value of z for 5% level of significance is 1.96, and the precision in this example d equal to 0.8. Now, apply the formula z equal to n equal to z square s square upon d square. So, z is the standard normal deviate or value of z is 1.96, s equal to 2.1 and d is 0.88. On solving this, we get the sample size is 20. So, the sample size for this study or this survey is 26. So, these are the two formula for estimation study. And now, we come to next another type of medical research, testing of hypothesis. In testing of hypothesis, we have two conditions. First is either we have when the proportion is the perimeter of our study, that means when we want to compare the proportion of two groups, then the sample size n equal to z alpha plus z beta whole square plus into p into q into 2 upon d square, where z alpha is the value of z for alpha level, that means first type of error, and z beta equal to z value for beta level, where beta is the second type of error, and p is the average percentage between two groups. Be remember, p is the average percentage between two groups and q equal to 100 minus p. D is clinical meaningful difference between two groups, or you can say margin of error, or you can say precision of our study. So, we take an example. Here we want to compare the two proportion. The above table shows data of two groups of patients either in low dose and high dose group. We have two groups one for low dose and another for high dose group. So, how many patients should be enrolled in the study to ensure that the power of the test is 80% to detect the difference in the proportions. That means, for we want to compare the proportion of two groups of high dose and low dose. A two-sided test will be used with a 5% level of significance. So, in this example, we say here the 24 patient in high dose group and 24 patient in low dose group, and in high dose group 4 patient has been died out of 24, while 20 were alive, and in low dose group 1 patient had died and 23 were alive out of 24, and the total patient in this study in this example are 48. So, first we calculate the proportion of high dose group and proportion of low dose group. So, the proportion of for example, we take P1 for high dose group, P1, and it will be equal to 4 upon 24 into 100. On solving this, we get 16.66 or 16.67, 666. So, it will becomes 16.67. Now, the proportion for low dose group, this is P2 and it is 1 upon 24 into 100, as 4 died out of 24. So, the proportion for high dose is 4 upon 24 into 100, and for low dose this is 1 upon 24 into 100, and it will equal to 4.2 in round off, and the total proportion is, if you add these, you will get 5 upon 48, but there is no need for to calculate this. So, we have two proportions 16.67 and 4.2. Now, first we calculate the value of P. The value of P is equal to P1 plus P2 by 2, as in I explain example formula, the P is the average of two proportion. So, it will be equal to P equal to P1 plus P2 by 2, and P1 we have already find 16.7 and P2 is 4.2. So, P is 10.4% and Q is, we know Q equal to 100 minus P, and it is 100 minus 10.4, this is 89.6%, and the value of Z alpha is 1.96 for level of significance 5%, for alpha 5%, the value of Z alpha is 1.96, and as the beta for 10%, when beta is 10%, then Z beta equal to 1.28. So, n equal to 1.96 plus 1.28, as the formula is n equal to Z alpha plus Z beta whole square into P into Q upon D square. This is the formula. On putting the value of these, we find the sample size is 125 in each arm. That means, we will take 125 in a high dose group and 125 in low dose group. So, this is when proportion is in our interest, then we use this formula, or you can say when we want to compare the two proportions. Second condition, when mean is the parameter of our study, then we use sample size n equal to Z alpha plus Z beta into S square into 2 upon D square. So, this formula, when we want to compare the mean of two groups, then we apply this formula. We remember, when we want to compare the two proportion, then we apply previous formula, and when we want to compare the mean of two groups, then we apply this formula. The sample size n equal to z alpha plus z beta into s square into 2 upon d square, where z alpha is the value of z for alpha error, z beta is the z value for beta error or second type error, s is the common standard deviation between two groups. So, in this we find the common standard deviation of both groups, and d is clinical magnificence or precision. For example, an investigator is planning a clinical trial to evaluate the efficacy of a new drug designed to reduce systolic blood pressure. The plan is to enroll patients and to randomly assign them to receive either the new drug or placebo. Systolic blood pressure will be measured in each participant after 12 weeks on the assignment treatment based on prior experience with the similar trial. The investigator expects that 10% of proportion will be lost to follow up or will drop out of the study. If the new drug shows 5 unit reduction in mean systolic blood pressure, this would represent a clinically meaningful reduction. How many patients should be enrolled in this trial to ensure the power of test is 80% and 90% to detect this difference? A two-sided test will be used with a 5% level of significance. Analysis of data from the Fragment Heart study showed the standard deviation of the systolic blood pressure was 19.0. So, the formula is z alpha plus z beta square into 2 into s square upon d square. So, if we take alpha 0.5 and beta 0.10, we get the sample size is 303, and if we put alpha is 0.5 and beta is 20%, here beta is 20. So, power is 80%, here power is 80%. So, we find the sample size is 226. So, in the if we change the value of beta, or then we find different sample size. Sample size n 1 303, and so 303 in first group and 303 in second group. If therefore, as the 10% patients are drop out, so we must enroll 10% of this in our study. The sample size n 1 303 and 303 will ensure that test of hypothesis will have 90% of power to detect a 5 unit difference in mean systolic blood pressure in patients receiving the new drug as compared to patient receiving the placebo. However, the investigator hypothesized a 10% attrition rate in both groups, and to ensure a total sample size of 303, they need to allow for attrition the number to enroll into percentage retained equal to desired sample size. Therefore, the n equal to 303 upon 0.9, it will be equal to 336. So, the investigator must enroll 306 participants to randomly assigned to receive either the new drug or a placebo. So, these are the different values of alpha and beta error for different. If we have alpha 0.5, then value of z is 1.96. If alpha is 0.1, the value of z alpha is 2.57, and if alpha is 0.001, then value of z is 3.29, and if type beta error is equal to 10%, then value of z is 1.28, and if value of beta is 15%, then value of z is 1.037, and if value of beta is 20%, then it will be equal to 0.842, and if we have value of beta is 25%, then value of z is 0.675. Now, another term is power of a test. The power of test is defined as 1 minus beta, or you can say it represents the probability to reject the null hypothesis when it is false, and the true mean is something other than the null value for the mean. So, power of a test is the probability to reject the null hypothesis when it is false, or you can say it is the probability to reject the null hypothesis when it is false. So, power of test is probability of a test rejects null hypothesis depends upon the difference of mean. So, if we take null hypothesis m1 and minus m2, that true power equal to probability of first type of error, that is alpha, and null hypothesis false, power equal to 1 minus type 2 error, that is 1 minus beta. The power of a test as a sample size increases, power increases; as population variance decreases, power increases; as the true mean difference increases, power increases. So, the formula for power is 1 minus beta. So, if beta is 20%, that means 0.2. So, this is 1 minus 0.2 and this is 0.8 and the power will be 80%, and if we beta take 10%, the power will be 90%. So, if you see in this curve, as the for n1 n2 25, for if we increase the sample size, power will be increases, as for if n1 n2 25, then power is this, and if we power n1 n2 is 100, then the power is 80%, and if m1 m2 the difference means between two sample is increases, then sample size is power is increases. Thank you.